So chapter 5 is on the cell membrane and its components and functions. Just as a reminder, majority of the cell membrane is made of phospholipids and cholesterol with some proteins involved. The phospholipid contains a hydrophilic part and a hydrophobic part. And within the membrane, to help make it a little less fluidity, a little less fluid-like, there are cholesterol is embedded in it. Cell membrane is selectively permeable, meaning there is a continuous boundary that selectively controls the flow of substances across it. The cell membrane has often been defined using what's called the fluid mosaic model, which suggests that the fluid membrane is a mosaic of components, primarily phospholipids, cholesterol, and proteins, that move freely and fluidly in a plane of the membrane. So cholesterol works to make the plasma membrane a little less fluid-like and stiffen it up just a little bit. And it has a four-ring interlocking form, hydrocarbon form. And in addition to it being part of the cell membrane, it is also the basis, as we learned from in chapter 3, is also the basis of all the steroids produced in the body. How does cholesterol stay in the membrane? How does it stay adhered in the membrane? The way that it stays adhered in the membrane is it has this hydroxyl group. Remember, this is one of those functional groups. It has this hydroxyl group that is hydrophilic and aligns with the phosphate groups of the phospholipids. And this is what keeps cholesterol tethered in the membrane. So remember, polar to nonpolar hydrogen bonding. A lot of different things going on here that keeps this cholesterol tethered within the membrane. It functions in the membrane partly to maintain the fluidity and keep things from becoming too slippery or to maintain some structure to the membrane. Also, it maintains the integrity of the cell membrane, and it helps secure important proteins in the membrane. The fluid mosaic model describes the interactions of the membrane components and how the lipid content helps the membranes to maintain fluidity and cell flexibility. We also have noticed from the fluid mosaic model that the higher the number of unsaturated hydrocarbons in the membrane, the more fluid-like that membrane becomes. And if you think about hydrocarbons being locked into their form when they become unsaturated, when they're saturated, they're less locked in their form. When things are less locked in their form, they're more likely to aggregate together easily. In that remember those hydrophobic interactions where we're working to exclude water? When they have the kinks in them, they are less likely to be able to aggregate well with one another, making the membrane more fluid-like. Moreover, there is a diffusion of lipids and proteins within the plasma membrane. Lipids and proteins can move laterally or sideways within the membrane, staying within the same layer. If they are not restricted by extracellular matrix interactions. These proteins and lipids can generally move within the membrane within one layer of the bilayer. Also, we can have transverse diffusion or flip-flop, where lipids on one layer of the membrane can flip to the other surface. Movement of lateral diffusion happens quite quickly. Transverse diffusion is much slower. It's a lot easier to stay within the one layer than to flip to the other layer. However, within the fluid mosaic model, we have to take into account that not all of the membrane proteins can freely move sideways because they are anchored to the extracellular membrane and cytoskeletal proteins on the inside. So on the outside, in the extracellular fluid, they're anchored to the extracellular matrix. On the inside of the cell, they're anchored to the cytoskeletal proteins. Also, there are glycolipids and glycoproteins within the fluid membrane. Remember, glyco means sugars or carbohydrates. So we have glycolipid molecules and we have glycoprotein molecules, or lipids that are attached to carbohydrates and proteins that are attached to carbohydrates. Their pattern, or the pattern of glycolipids and glycoproteins in a membrane, can serve as a fingerprint in cell identification. So, pattern of the glycolipids and glycoproteins on one specific cell can allow for that cell to be identified as a self cell, meaning it belongs to that body, or if it's not identified, it's considered foreign. An example of this that you've probably at least heard of are transplant rejections and why we have A, B, and O blood groups. Also, we can have all animal cells have a glycocalyx, which is where glycoproteins make up a sugar coat. This coat functions in protecting the cell, recognition of signaling molecules, and cell-to-cell adhesion and cell-to-cell recognition. So cells can use this to adhere to other cells, can also use this to be involved in cell-to-cell recognition. Lipid rafts exist in all eukaryotic cells, and are mostly in the plasma membrane. Lipid rafts are enriched with sphingolipids, and another type of lipid, sterols, remember cholesterol, so a derivative of cholesterol, many derivatives of cholesterol, and these rafts, with being enriched in sphingolipids and sterols, they are depleted in phosphoglycerol lipids. They have a very ordered composition where they can include certain things and exclude other things. It is hypothesized that these rafts that are what you'd call patches on the membrane are involved in signal transduction pathways, cell trafficking, and host pathogen relationships. With regards to cell membrane proteins, we have integral membrane proteins or intrinsic proteins, where the proteins have one or more segments that are embedded in the phospholipid bilayer. These can be classified as transmembrane proteins also. Transmembrane meaning they span the membrane. So they can either be embedded in the phospholipid bilayer, or they can also span the membrane. Transmembrane proteins span across the phospholipid bilayer. They consist of one or more membrane-spanning domains. Residues with hydrophobic side chains interact with the fatty acid groups of the phospholipids, by which they are anchored in the membrane, with their hydrophilic linking chains being observed on either side of the membrane. They can be made of domains possibly up to several hundred residues that extend into they can have domains that are up to several hundred residues long that span into the aqueous medium on each side of the bilayer. So hydrophobic residues embedded in the membrane, hydrophilic residues that flow into the intracellular fluid and extracellular matrix and fluid. In addition to having integral proteins, we have peripheral membrane proteins or extrinsic proteins. These are proteins that do not interact with the hydrophobic core of the phospholipid bilayer. They are usually bound to the membrane indirectly by interactions with integral proteins or directly by interactions with lipid polar head groups. I have hydrogen bonds here. The hydrophilic ends of this transmembrane protein interacting with this peripheral membrane protein. Or interacting with the phospholipids, polar heads. with ionic and hydrogen bond interactions. Membrane proteins can have lots of different functions. For example, we have channel proteins that allow the passages of molecules through the membrane via a channel in the protein. We have carrier proteins which combine with the substance to be transported and assist the passage of molecules through a membrane. We have cell recognition proteins, glycoproteins, that help the body recognize foreign substances versus substances that are self-proteins, cells. Receptor proteins that bind with specific molecules and allow a cell to respond to signals from other cells. Enzymatic proteins that carry out metabolic interactions directly, and junction proteins that attach adjacent cells together. So we have transport proteins, protein that provides a hydrophilic channel across the membrane that is selective for a particular solute. Some of these transport proteins require ATP to actively drive substances from a high concentration to a low concentration. We'll talk about active transport here in a moment, which is what that is. We have receptors for signaling transduction, which bind to a chemical messenger out in the extracellular fluid that can cause a change in the shape of the protein that will initiate a chain of chemical reactions within the cell. We have components of the cytoskeleton and other components of the extracellular fibers that anchor membrane proteins, which helps maintain the cell shape and location of certain membrane proteins. So cells that can attach to the cytoskeleton and the extracellular matrix. We have enzymatic proteins that serve as an enzyme with their active site, which we will talk about in the next chapter, exposed to substances in the adjacent solution. We can have teams of these enzymatic membrane proteins that catalyze a sequential series of steps in a metabolic pathway, which we will talk about in the next chapter also. We have intracellular joining proteins or cell adhesion molecules, or CAMs, that provide temporary binding sites that guide cell migration and cell-to-cell interactions. We also have glycoproteins, like we've talked about, that serve as identification tags that can be recognized by other types of cells. A specific type of membrane proteins that we have that we've touched on, the cell adhesion molecules and proteins that are involved in cell-to-cell interactions and extracellular adhesion interactions. Within that group, we have specific proteins that are specific for adhesion to cell, from cell to cell, and communication between cells. These are junctions between cells. that can allow for adhesion, meaning intercellular filaments between the cells. Some examples are desmosomes, which are intercellular cytoplasmic plaques, which we talked about in the last chapter. It was very the end of the chapter 4, where the intermediate filaments interacted with the desmosome plaques. So this is where we saw that the cytoskeleton proteins are linking with the desmosome plaques to help hold cells together. Tight junctions that form waterproof, impermeable barriers, keeping when we have a flow, when we have a cell lining, and we require that the materials flow through the cell and not in between the cells, tight junctions are there to form these impermeable waterproof barriers. We also have gap junctions that are plasma membrane channels that join cells and allow for communication between those cells. These are vitally important in heart muscle function and smooth muscle function. These are little channels that allow the movement of solutes and ions in between cells to allow for those cells to communicate with one another. With the cell signaling of membrane proteins, it works in a process where a receptor, that membrane protein, binds to a signaling molecule. When it binds to a signaling molecule and or a ligand, that membrane protein becomes activated, and it can activate other proteins within, or enzymes or proteins within the cell, leading to a transduction of a series of proteins that end with other proteins being activated. So a ligand binds to a receptor, which is the membrane protein. And, and when that ligand binds to the membrane protein is then activated to be able to activate a series of proteins that ultimately will result in a response where a targeted protein that has been activated will bring about either a change in shape or movement of the cell, altered metabolism or cellular function, or altered gene expression and or the types and amounts of proteins that are produced. So ligand binds, you have a transduction pathway where a series of proteins results in a protein that is activated, and the specific protein that's activated then performs a cellular response. And that cellular response can either be an altered shape or movement of the cell, altered metabolism or cellular function, or an alteration in the gene expression and the types and amounts of proteins that are produced. So the phospholipid membranes show a selective permeability, where gases, hydrophobic molecules, and small polar uncharged molecules can diffuse through. However, large polar molecules and charged molecules and ions are not allowed to diffuse through. Diffusion requires a concentration gradient. And a concentration gradient is defined as the difference or concentration of when there is a difference or in the concentration of molecules or ions, aka solutes, in one particular region than the number in another particular region. So where there's a difference in the concentration of molecules or ions in one region versus the number in another region. In the absence of other forces, substance, so in the absence of any other energy form, a substance moves from a region where it is more concentrated to a region where it is less concentrated. And this is just based on the own vibration kinetic energy of the molecule itself. No extra energy is required. If you think about the example that I gave you about the fact that if we have 400 people in a room that's probably like 20 feet by 10 feet, and then we have in an adjacent room of the same size that we have 5 people in that room, and we have this one door that connects the two regions. How many of us are going to try to get into that other room? Probably a good majority of us, because everything is without any other external forces being involved. Everything will move from a high to a low concentration going down its gradient. This passive or simple movement down a gradient does not employ ATP. The examples that we're going to discuss, or the processes that we're going to discuss of passive transport, are filtration, diffusion, facilitated diffusion, and osmosis. The active transport mechanisms that we're going to discuss are primary active transport, secondary active transport, endocytosis, and exocytosis. So this passive transport, as we were discussing about a couple seconds prior, is this movement of ions or other atomic or molecular substances across a cell membrane without the need of an energy input. So it does not require the input of cellular energy, because unlike active transport, it is driven by the natural tendency of a system to move from a more organized area or a more concentrated area to a less concentrated area or less organized area. This is called entropy, which is the natural tendency of things to move toward disorder. So this passive transport is driven by the natural tendency of molecules to grow in entropy or grow in more disorder or more diffuse. The energy that drives passive transport is this kinetic energy, or the natural inherent energy of molecules to move. It is used to do the work of transporting the solute down the concentration gradient. So this is an inherent kinetic energy of the molecules to move. Filtration is driven by physical pressure that forces fluid through a semi-permeable membrane. For example, the primary driving force of fluid transport between capillaries and tissues is a hydrostatic pressure, so the pressure of the water. And this hydrostatic pressure is the pressure of any fluid enclosed in a space. Thus, fluid generally moves out of the capillary and into the interstitial space by a process known as filtration. If you picture your hose, you have a garden hose, and you have liquid flowing in it, water flowing in it. The pressure of the water molecules bouncing along the garden hose as they rush from one end of the hose to the other end is the hydrostatic pressure. And when a membrane is semi-permeable to water, we get filtration. So if our hose was semi-permeable to water, we would get movement of water out of the hose, just based on the hydrostatic pressure within that hose. So filtration is when a physical pressure forces fluid through a semi-permeable membrane. We see this in capillaries, where fluid moves through in between little fenestrations or clefts in between the cells when fluid moves into the interstitial space through filtration. We also see this when we are working to filter things with our kidneys. The net filtration pressure in the kidneys is a combination of the outward pressure, the water pushing out, and an inward pressure to bring things back in. Depending on which pressure is higher, will determine how much is filtered out. If the hydrostatic pressure is higher than this osmotic pressure, then we see that things move out, and filtration, the net is a movement of things to move out or filter out of the capillaries. Diffusion is a net movement of solute down a concentration gradient. Although molecules can collide randomly, the net movement is away from the place with the most collisions. So for example, of the 400 people grouped in one little area right here, going to be touching a lot and probably not very comfortable. You slowly move out to get away from where we have so many collisions, and move and diffuse out until the concentration is uniform across the space. It's a passive process. Sneezing, moving out from a concentrated area to a less concentrated area. Things always move from a high concentration to a low concentration inherently. An example of this, of all things, fluid, ions, solutes, everything will move from a higher concentration to a lower concentration. One example of this is gas exchange in the lungs, which occurs by diffusion, where higher oxygen concentration within the alveoli of the lungs will diffuse into the capillaries just based on the fact that there is a lower oxygen concentration in the capillaries. Same thing for CO2. It will diffuse from the capillaries, where it is a higher concentration of CO2, to back into the alveoli where there is a lower concentration of CO2. Factors that affect the diffusion rate is the steepness of the concentration gradient, the magnitude of it, the molecular size. The smaller the molecules, the faster the diffusion, the temperature. The higher the temperature, the faster the diffusion. Membrane surface area. The more surface area you have, the more diffusion you will have. Remember, we talked about how this was involved in determining cell size. Membrane permeability and pressure. Facilitated diffusion is when molecules move across the plasma membrane with the help of a channel or carrier protein. So this is when there's a flow of solutes through the interior of passive transport proteins down their concentration gradients. These passive transport proteins allow proteins to move in both ways. But remember that passive, there's that inherent drive to move from a high to a low. This facilitated diffusion does not require any energy input because it is driven by the inherent kinetic energy of the solutes and molecules themselves. Facilitated diffusion is when transport molecules are specific to the molecules that are transported within them. Channel proteins are capable of transporting proteins at a very high rate compared to carrier proteins. Carrier proteins have to undergo a change in structure. Channel proteins do not. Change in structure requires half a second longer. So channel proteins are much more efficient. The main difference between channel proteins and carrier proteins is their mechanism of transporting the molecules across the membrane. One requires a structural change, the other one does not. Osmosis is a special case of diffusion that focuses on solvent water movement rather than solute movement. This is the diffusion of water across a selectively permeable membrane, where the solute concentration on one side is high, but the water concentration is low. If you remember when we talked about this in this last class period that we had, if the solute concentration is higher in a given certain limited volume or capped off volume, the water concentration has to be lower there. Alternatively, if the solute concentration is low, the water concentration has to be higher. When we're looking at osmosis, water can diffuse in both ways across a membrane, but the solute is confined and therefore the membrane is impermeable to the solute. In a case of like solutes, non-water things, the water is going to move towards the lower water concentration. So there is also with this, there is a pressure that will develop that's due to the osmosis or the incoming flow of water into the area of the higher solute concentration. And this is called the osmotic pressure. So we have this experiment using a U-tube, a U-shaped tube, where we have one side of the membrane is pure water. The other side of the membrane has some solute in it. Because we know that the side that has solute in it has to have the lower water concentration, water is going to flow from the side that's pure water to the side that's got the solute in it, because that's the lower water concentration. And this movement of the water continues because of the hydrostatic pressure of the water to move from the higher concentration into the lower concentration. We can apply a pressure to the solution with the solute in it that can block how much water is moving into the side with the solute in it. And so if we apply enough pressure, this osmotic applied pressure, if we apply enough pressure, we can move the water back from higher concentration from the lower concentration into the higher concentration. This is called the osmotic pressure. So this is the pressure within the liquid with the solute in it that resists the flow of incoming water. When the osmotic pressure equals the hydrostatic pressure, we can restore things to the starting conditions. Tonicity is the relative concentrations of solutes that determine the direction and extent of diffusion. Tonicity is commonly used when describing the response of cells immersed in an external solution. Tonicity is relative, or the concentration of a solute may be higher on one side of a membrane relative to the other. It's always relative to what's inside the cell. Isotonic solutions are when the solute and the water concentrations, when the solute concentration inside the cell and the solute concentrations outside of the cell are equal. And what iso meaning the same, tonic meaning the number of solutes. In the case of a cell being put in an isotonic situation, because the concentration of water is equal on both sides, there is no net gain or loss of water into the cell or out of the cell. In the case of hypotonic solutions, the concentration of the solute in the external fluids is lower than that inside the cell. Cells that are placed in this hypotonic solution, meaning that the water concentration is higher on the outside, will undergo a swelling because the concentration of water is higher outside of the cell. Water will work to move into the cell. This results in what's called turgor pressure in plants and may cause hemolysis in animal cells. When a cell is placed in a hypertonic solution, meaning that the solute concentration is higher in the external solution rather than inside the cell, the cells will shrink. In the case of plant cells, this is called plasmolysis. In the case of animal cells, this is called crenation. So in a hypertonic situation, the net flow of water will be out of the cell because the solute concentration is lower out. Solute concentration is greater outside, so the water concentration is less. In the case of the hypotonic situation, the solute concentration inside is more, so the water concentration is less, so water moves into the cell. In the case of the isotonic situation, there is no net flow in or out of the cell with regards to water. So to reiterate this, a normal red blood cell put into an isotonic situation means that the solute concentration is equal inside and outside of the cell, and there will be no net movement of water. A cell that's placed into a hypertonic solution, meaning that the solute concentration is much higher outside of the cell, the flow of water from the inside of the cell will flow out of the cell, causing the cell to shrink or undergo crenation. When the cell is put in a hypotonic solution, the solute concentration is greater inside the cell than outside of the cell, and the net movement of water will be into the cell, which can cause hemolysis of the cell. Lysis meaning to blow apart, so hemo meaning blood, hemolysis. This is why plants their structures are the way they are. For example, dehydrated celery contains many dissolved molecule solutes relative to the water molecules. When it is placed in a salt water, meaning a hypertonic environment, the water will move out of the cells into the salt, causing shrinkage of the plasma membranes inside the plant walls. It's called plasmolysis. When wilted celery is placed into distilled water, you've thus placed the plant in a hypotonic environment, where the water will move into the cell, pressing the plasma wall up against the cell membrane, causing turgor pressure. So plasmolysis, the cell membrane is shrinking within the cell wall. Turgor pressure, the cell membrane becomes tightly adhered and is bulging and putting pressure on the cell wall. Turgor pressure, putting pressure on the cell wall. So comparison of animal and plant cells. Hypertonic plasmolysis crenation. Isotonic turgor pressure. Hypotonic hemolysis or normal. What environment do animal cells prefer? Just looking at this, what do they prefer? They prefer this. We talked a lot about how structure is related to function. We want our cells to maintain their regular structure, because that ensures that they'll maintain their regular function. In the case of plant cells, they like the hypotonic environment, where there is a pressure of the plasma membrane to adhere up against the cell wall. How would this explain the preservation of beef jerky? This is hunting season, and people will often preserve meat by adding extra salt to it. Why do you preserve it that way? You think about it, bacteria that possibly decide that that's where they want to reside on that meat, when they try to survive on that meat, you put them into a hypertonic environment. And at that point, they're going to shrink and die. So salting meat causes any bacteria that decide to reside in that meat in a hypertonic environment. One other question, if water is polar, how does it move through the plasma membrane? Two choices. It can move through with aquaporins, or it's small enough that with the random fluctuations in the fatty acid tails, it can slip through the plasma membrane. So with aquaporins, either random move fluctuation, which happens a little bit, or through the insertion of aquaporins, aqua meaning water, porins meaning a pore. So water can diffuse through the membrane because it's small. So some molecules can slide through the non-polar phospholipid tails. However, osmosis can also be facilitated with the insertion of water channels or aquaporins, meaning a facilitated diffusion of water, osmosis. Remember, the only difference between solute diffusion and osmosis is that in osmosis the water is being diffused and not solute. So osmosis is a diffusion of a solvent. Primary active transport is where the net diffusion of a solute is against its concentration gradient. So we talked about that room where we are in a room that's 20 by 10 feet, and there's 400 people in that room. Well, the majority of us are going to try to get out of that room. But what if there is someone at the door who keeps pushing people into the room that has 400 people in it? That is what we call active transport. The person or the protein that regulates that door and keeps pushing people into this much more higher concentration is activated usually by ATP in the form of phosphate. It can also be something we might talk about later in this semester, guanosine triphosphate. But usually it's adenosine triphosphate. So the activated protein is activated by phosphorylation by the addition of a phosphate group. ATP gives up the phosphate that activates the protein, so energy is required. The binding of ATP changes the shape of the protein, and and thus changes the affinity of the channel protein, carrier protein, for the solute that's going to be transported. Probably one of the more famous examples and most well-used examples of primary active transport is the sodium phosphate ATPase pump. In the case of this sodium phosphate ATPase pump that pumps sodium from a lower concentration inside the cell to a higher concentration outside of the cell. And with each pump being motion, it also transports 2 potassium from the outside of the cell into the inside of the cell. And at this point in science, we understand the process by which this occurs. So in step 1 up here at the very top here, 3 cytoplasmic sodium bind to a pump protein. The protein is in a configuration where it is open to the inside of the cell, and 3 sodium bind. When the 3 sodium are bound, this promotes the hydrolysis of a bound ATP. And the energy released during this reaction phosphorylates this pump. So that hydrolysis, the ADP, adenosine diphosphate, leaves, and there's a phosphate attached to the carrier. This binding of the phosphate changes the configuration of this protein, carrier protein, so that it becomes open to the other side, to the extracellular side. And at that point, the sodium are released. And at the same time that now the cell is open to the outside, 2 potassium bind to the pump. The phosphate then is released. This binding of the potassium to the pump triggers the release of the phosphate group, which is accompanied with the configuration of the channel protein switching to where the protein is now open to the inside of the cell, and the potassium are then released. The pump then binds another ATP. So just to reiterate here, we start with this carrier protein being open to the inside of the cell, and there is an ATP bound to it. 3 sodium bind. The binding of the 3 sodium triggers the phosphorylation of the hydrolysis and phosphorylation of the carrier protein. With that hydrolysis, the carrier protein undergoes a conformational change where the sodium is released to the outside At that point, we're open. The cell is open to the outside, so two potassium bind. When the two potassium bind, the phosphate group is released from the carrier protein, and there's a conformational change where the potassium are now released to the inside of the cell, and then another ATP binds to the pump, and the cycle goes again with sodium binding again from the inside. Another form of active transport is when a transporter protein couples the movement of an ion down its concentration gradient to the uphill movement of another molecule or ion against its concentration gradient. So when the movement of one molecule down its concentration gradient is coupled with the movement of another ion against its concentration gradient. So when we use the energy of pumping one molecule against its concentration gradient to drive the movement of another molecule, this secondary active transport depends on the ion gradient that is created by primary active transport. So we have to generate a concentration gradient first by primary active transport. The potential energy that's stored in these gradients indirectly drives the transport of other molecules. For example, a low sodium concentration maintained inside the cell by the sodium-potassium pump that keeps driving sodium out of the cell strengthens the sodium's drive to enter the cell. Sodium can be used to drag other molecules along with it when it flows into the cell using carrier proteins, usually symporters, meaning they're moving in the same direction in the membrane. Some molecules, amino acids, and ions are usually transported into cells via this secondary active transport. So, secondary active transport is driven by the concentration gradient created by primary active transport. So we have this ATP-driven sodium-potassium pump that keeps the concentration of sodium low in the cytoplasm. By keeping the concentration of sodium low inside the cytoplasm, by maintaining a higher concentration in the extracellular fluid, we have a potential energy source in that the sodium wants to move to a lower concentration. Inherently, it wants to move. So we can use this movement of maintaining this high concentration of sodium, we can use it to drive the movement of other molecules into the cell. So in the case of the sodium glucose symport transporter system, as sodium diffuses back across the membrane through a protein, that protein can co-transport glucose against its concentration gradient into the cell. So we can move by allowing sodium to move down its concentration gradient, we can drive the movement of glucose against its concentration gradient by dragging it along when we move sodium down its concentration gradient. So we maintain the we build up the concentration gradient here, and we use that concentration gradient as an energy form to drag sodium from a lower concentration to a higher concentration. Another type of diffusion that is vital in biological systems is a membrane potential or transmembrane potential or a membrane voltage. This is similar to your regular average Duracell battery in that we have a separation of charge by which the flow of charge across a membrane can be coupled to form the work of something else. So in the battery, there's a flow of charge from one area of the battery to another area of the battery. With regards to a membrane potential, there's a separation of charge or electric potential across a membrane. So a difference in the charge between the interior and the exterior of the cell. When we have a difference in charge, just like when we were talking about polar molecules and nonpolar molecules, like a polar molecule, when there's a separation in charge across that membrane or molecule, we refer to it as being polarized. Factors that contribute to the membrane potential are an unequal distribution of ions across the plasma membrane, an inability of most anions to exit the cell, and the electrogenic nature of the sodium-potassium pump. So this is that ATPase pump, this third one. The typical membrane potential for an unexcited cell ranges from minus 40 to minus 80 millivolts, meaning that the inside of the battery or the inside of the cell is more negative than the outside. So there, within every cell, there is this resting membrane potential, which is the same as a membrane voltage, meaning a separation in charge across the membrane. Endocytosis, endo meaning into, cyte meaning cell, is this process of capturing a substance or particle from outside of the cell by engulfing it with the cell membrane. Phagocytosis is when a large solid material is taken in by endocytosis. So phago meaning to eat, cyte meaning to cell. So this is cell engulfing, cell eating. An example of this is when human white blood cells engulf debris or viruses. Pinocytosis is like cell drinking, when tiny pinches of a liquid or very small molecules are being taken into the cell. I always think of pino as meaning pinching, so little tiny pinches, rather than phagocytosis where it's just like, and pinocytosis is just this very small pinching of liquid or very small particles and bringing them into the cell. Receptor-mediated endocytosis is a very specific form of pinocytosis using receptor proteins and a coated pit. So phagocytosis is when the cell engulfs a large particle by forming pseudopods around it and enclosing it within a membrane sac called a phagosome. The phagosome is combined with the lysosome. Remember, that's the destructive organelle within the cell. Pinocytosis is when the cell gulps or just swallows small drops of the extracellular fluid. These two forms of endocytosis are non-specific. Receptor-mediated endocytosis requires that something binds to the receptors, meaning those membrane proteins we were talking about earlier. Extracellular substances bind to specific cellular receptor proteins, enabling the cell to ingest and concentrate specific substances in protein-coated vesicles. The ligands may simply be released inside the cell or combined with a lysosome to digest the contents. So they can either be just transported to the other side and released, or those vesicles can combine with the lysosome. And the receptors are then recycled back to the plasma membrane. Exocytosis is where intracellular vesicles full of product are produced in the Golgi apparatus that then fuse with the plasma membrane and secretion occurs. So exocytosis meaning to move something out of the cell or have it exit the cell. Exocytosis requires the fusion of a secretory vesicle with the plasma membrane. So a summary of these transport mechanisms. Passive transport, filtration, diffusion, facilitated diffusion, carrier-mediated or channel-mediated, and osmosis. Remember, that's just the inherent kinetic movement of a molecule to move from a high to a low concentration. In the case of active transport, we can have primary active transport where the hydrolysis of ATP is being directly used to move one solute from a low concentration to a higher concentration. Or we can have secondary active transport, where the primary active transport of one solute is used to develop a high concentration of that solute that can drive the movement of another solute from a low concentration to a higher concentration. Also, active transport includes these vesicular transport movements, like endocytosis, that includes phagocytosis, pinocytosis, receptor-mediated endocytosis, and exocytosis. Last thing that I want to hit on with this chapter is animal extracellular matrix, which is a meshwork of proteins and polysaccharides that are in close connection with the cells that produce them. So this is something secreted by cells. 5 major things are observed in animal extracellular matrix. They are collagen, which helps resist stretching; elastin, which provides a resilience and a flexibility to the extracellular matrix; fibronectin, which is an adhesive protein; integrins, which play a vital role in cell signaling; and proteoglycans. Remember, protein meaning protein, glycan meaning carbohydrate. So proteoglycans attach a long centrally placed polysaccharide to cell membranes and resist compression of extracellular matrix. Proteoglycans can also assist in cell signaling by regulating the passage of material through the extracellular matrix to the plasma membrane. So we have the actin filaments of the cytoskeleton that interact with proteins such as integrins. So a transmembrane protein can be part of the extracellular matrix and be an membrane protein. And these components in the extracellular matrix, collagen, proteoglycans, elastin, and fibronectin, can interact and adhere to the membranes, the cells that produce them. Just to hit on one more thing, talking about plant cell walls. Plants have a really permeable cell wall that's made of cellulose as the main component. There are plasmodesmata. We talked about desmosomes. These are cell junctions within plant cell walls that penetrate the cell wall. Each contains a strand of cytoplasm, which allows for the passage of materials between the cells. And these are little channels, similar to gap junctions in animal cells. Cells in woody plants have a secondary cell wall, which contain lignin and more cellulose fibers than the primary cell wall. So some woody plants have two cell walls rather than just one.